Viral vectors are tools used in human gene therapy with a promise to treat or even cure genetic disorders such as beta thalassemia, cystic fibrosis, and hemophilia. We can essentially think of viral vectors as biological packages containing therapeutic genes that could replace faulty genes in patients with genetic disorders. Chemical engineers play a crucial role in developing scalable, reproducible, and robust processes that could produce clinically relevant quantities of viral vectors. For example, to treat one patient with beta thalassemia, a blood disorder, 3.5 times 10 to the power of 11 viral vectors are needed. This quantity has the same order of magnitude as the distance from the Earth to the Sun. To achieve manufacturing goals, chemical engineers must move along an understand, predict, and control axis. Let's take a closer look at the production bioreactor where the viral vectors are produced and how we can apply these engineering design principles. A bioreactor is a vessel in which biological species are grown, maintained, and manipulated in a controlled environment for biologics manufacturing pipelines. To simplify things, we can think of nutrients and transfection reagents as our reactants, cells as the catalysts, and viral vectors as our products. As you might expect, we also have undesirable byproducts which can have detrimental effects on cell growth and viral vector production. To understand biological reactions inside our vessel, mass balance equations using a set of ordinary differential equations have been used to model our system's dynamic behavior. Here, the reaction kinetics are based on the modified Monod equation. We can even rewrite these equations in a more compact and elegant form shown here. To validate our model and increase the accuracy of their predictions, we have fitted experimental data from the literature using weighted least squares technique to determine parameters in our system of ODEs. Essentially, this becomes a multivariable error minimization problem to find a good fit. Let's now take a step back to understand what the cell culture model tells us. Critical process parameters that appear in our model include pH, temperature, dissolved oxygen, and agitation rate. Our model provides a relationship between these process parameters and our response variables, which include production runtimes and product concentration. We can then vary the process parameters and use our model to predict and optimize the system's response. Ultimately, the fundamental process parameters can be closely controlled in large-scale manufacturing pipelines to produce viral vectors. In summary, canonical biochemical engineering principles must be applied to minimize the knowledge gap in viral vector manufacturing and expedite human gene therapy's clinical translation using viral vectors.